In this episode of Brain Ponderings, my guest is Allan Herbison. He's a professor at the University of Cambridge. He's a neuroendocrinologist. So he studies how the brain and circuits in the brain control endocrine organs, and specifically he focuses on reproductive organs, ovaries and testes. And there's been a lot of elegant work done recently by him and by others on understanding how the brain at the cellular level, how certain neurons in the brain, by releasing certain neurochemicals, control the gonads, control the estrous cycles, control ovulation, even control the onset of puberty. And this is very important information, not just from a general science standpoint, but there are translational implications in terms of developing treatments for infertility and other disorders of reproduction. So Allan, you're a native New Zealander, is that right? You grew up in New Zealand?
Yep, I've spent most of my career bouncing backwards and forwards between New Zealand and Cambridge. Yep.
Yeah, and you have both a medical degree and a PhD, is that right?
Yeah, in New Zealand, I went through the medical course there and got a medical degree and worked for a little bit, and then was lucky enough to get a PhD to come to Cambridge to just to see what life was like as a discovery scientist. So I did a PhD, and essentially I never looked back. So I've continued that. I've spent a career in biomedical research, looking at how the brain controls fertility. So it's been a really lucky guy.
And was it at Cambridge in your PhD work where you first started working on neuroendocrine systems?
Yeah, not really actually, because in the course of my medical degree, I had the opportunity to take a year out and do some. And for me, it was neuroscience, because it was always neuroscience. And I went to a lab, and they said, "Oh, well, you should be working on this part of the brain." And in fact, my very, very first experiments that I ever did were on the arcuate nucleus of the hypothalamus. And here I am, nearing the end of my career, working in the arcuate nucleus of the hypothalamus. So you might say not very imaginative, but I plowed a very, very narrow but probably quite deep furrow in my career.
Now, in the history of endocrinology, like the early experiments were kind of straightforward. And I actually, when I was working on my PhD at University of Iowa, I was a teaching assistant in a laboratory, an endocrinology laboratory, where we would remove the ovaries from a female, then take the blood and measure something, estrogen, for example, or we'd remove the testes or the adrenal gland and so on. And so that was kind of the classic early experiments in kind of establishing that these organs in the periphery are like critical for a lot of things. So you're working on how the brain controls the gonads, but I think it's instructive for listeners and viewers to know just a little bit of basics about the gonads. People have heard of estrogen and testosterone, but where do they come from?
Yeah. So I mean, obviously, we're dealing with ovaries and testes, and the ovaries is the principal organ that's out there that's producing estradiol and also progesterone, which are the two key gonadal steroid hormones that are really setting the body. Many, if not all, organs are responding to these hormones, and it's setting the tempo across the menstrual cycle. Whereas for males, it's primarily testosterone. I mean, we should remember that females also make testosterone, and males also make estrogen and a small amount of progesterone, but there's just a tenfold difference in the amounts that are produced. So the testes is producing testosterone, and it's more or less operating by itself as a single hormone modulating the body and feeding back onto the brain. But alongside that, we have to remember that the male is a simple creature. It doesn't exert reproductive cycles. We're just rather continuously active and possibly don't even go through an equivalent of a menopause as such. So we just start up at puberty and we just sort of keep going. Whereas females have to do much more clever things like having, ovarian cycles, and that requires estrogen and progesterone to coordinate a whole variety of different, body organs. So it's a little different, for males and females, but the concept is generally the same. That we have gonads that are, I didn't mention, of course, making gametes for actual reproduction, but in terms of body physiology, are making their own gonadal steroids.
And so the way the brain controls the gonads and say the adrenal glands is there are neurons in certain brain regions, and you mentioned the arcuate nucleus of the hypothalamus, and then these neurons actually, they can release neurochemicals and affect what's called the pituitary gland. Now, how does this hypothalamic pituitary system control, say, estrogen production?
Yeah. So a long time ago, it was in the early 1900s. I mean, there's a guy, Harris, who was here in the UK, Geoffrey Harris. He was the guy who's credited with really establishing experimentally that the link between the brain and the pituitary gland was a neurohumoral system. In other words, that neuropeptides or transmitters were being released at the very base of the hypothalamus, and these were then flowing down in the bloodstream in the portal, what's called a portal circulation, to actually act in the pituitary gland. So in the context of reproductive of control and fertility, the key players are a neuron called the GnRH neuron, gonadotrophin releasing hormone neuron. So its nerve terminals really poke out into the base of the brain, and they release this molecule GnRH that passes down the portal circulation and then acts in the pituitary. So in the pituitary, there's a specialized cell called a gonadotroph, and GnRH acts on that, and the gonadotroph in turn then releases two hormones, luteinizing hormone and follicle stimulating hormone, and then they pass through the circulation down to act on the ovary. And so, as their names sort of imply, I mean, follicle stimulating hormone is the one that's primarily involved with developing the follicles, so the developing follicles that are sitting in the ovary that are waiting to grow. It's very important for that. And LH is the luteinizing hormone, also has a role in that, but its main claim to fame is that it's the thing that actually triggers ovulation, that makes the follicle actually ovulate, so that the egg is released actually into the uterine tube. So that's called luteinizing hormone. So we have these hormones that are being.
So the LH and FSH are always released at the same time.
Yeah, so we have one molecule, GnRH, controlling the gonadotroph release of LH and FSH. Where we get a little bit of differential control is that, I was talking before about the hormones that feed back, estrogen and progesterone. So as in with any sort of classical physiological, homeostatic circuit, these hormones feed back to control, the release of the molecules that are regulating them. And so they act both at the pituitary gland and they also act back at the brain. Nice. And so there's probably there is some differential feedback control of LH and FSH release. So, a key thing is that these hormones are released in a pulsatile fashion. So when LH is released in a pulsatile fashion, then we'll also get FSH released. When we have an LH surge, we also have FSH. But the actual amounts of it can vary differentially depending on the feedback of hormones and other things like inhibins and such like.
And the estrogen and progesterone, so they act, there's receptors in the cells that are actually affect gene expression. They're transcription factors.
Yeah.
So these cells in the pituitary that produce FSH and LH, they have receptors for both estrogen and progesterone. Is that right?
That's right. And androgens. Yeah. So there's quite effective feedback at the pituitary in terms of the control of the system. And in a way, I think one of the things that we've grappled with, have not resolved, but grappled with, is the degree of feedback. You know, how much feedback is there at the pituitary gland as opposed to feedback at the brain to control the release of GnRH? And is that really different in different mammalian species and stuff? Those sorts of questions. I mean, there is a line of thinking that in humans, for example, that feedback at the pituitary is actually the most important factor, whereas you say certainly in rodents and probably also sheep, that's probably not the case. So you know, these are quite quite meaty issues that have not really been resolved.
I see. And then, so the GnRH neurons, I was fascinated. I looked back at your work on them, and so my work focused on, you know, higher brain regions, hippocampus, cerebral cortex, and a lot of work in culture, but also in vivo. And the neurons there, they're classic neurons. They have a cell body, a long axon that often extends a long distance, and then dendrites. And the dendrites receive the information, so all the synapses are on the dendrites. There's no synapses on the axons, but I was fascinated by what you've dubbed the dendron in these GnRH producing neurons. Can you describe that? Because I think it's really kind of a neat oddity, really.
It is, and I think we have to tip our hat to the GnRH neuron itself as being a complete oddity. You know, a really bizarre, and a really quite bizarre situation when you consider that, you know, every species requires GnRH neurons to continue, is that these neurons developmentally are extremely unusual in that they're actually born in the nose. So, you know, all other neurons, respectable neurons, are born in the brain and, you know, migrate and develop in a particularly normal way. But the GnRH neurons, they're actually born in your nasal placode, and this is for all vertebrates that have been looked at. And over the course of embryogenesis, they actually have to migrate from the nose and enter into the brain. And then once they're in the brain, they then have to migrate down into the hypothalamus. So this is just a remarkably odd system. And of course, creates with it many, many dangers. And you know, a rare human situations where individuals that you know, they don't go through puberty and they're infertile, and you know, they walk into the doctor's surgery, and the doctor, if he's switched on, will say, "What's your sense of smell like?" And often these people won't smell either. So actually, they've had some developmental defect in their olfactory part of their brain, which has meant that their GnRH neurons are actually still stuck in their nose. So not only do they not smell very well, but they also, unfortunately, are infertile. So sorry, I digress. But you know, the GnRH neurons are really, you know, right from the start, they're a very, very odd type of cell. So,
Allan, you know, so what about from an evolutionary perspective? So as you know, lower mammals, you know, you've used rats and mice. We did, you know, there's some other species, but the sense of olfaction is really important in their not only survival, but I think in their reproductive behaviors, right? Do you think there's any potential evolution? Well, there has to be an evolutionary explanation, but
yeah, I think the best thing that's been put forward is that there was a I forget the name of the organism. I should remember, but it, pre-vertebrate. It's a tunicate. Sorry, a tunicates.
Huh.
And what they do is they have an organ that actually sits out floating in the seawater, and not only is it the olfactory neuron or olfactory or sensing organization, it's also the thing that's responsible for reproduction. And so we think, and it makes a form of GnRH. So it seems that early in development, there was one cell that was doing olfaction and being responsible for reproduction. And then what's happened is that with, that cell is basically split, and one has been left doing the olfaction, and the other one has actually moved along as we develop brains and pituitaries and such like, and moved in that direction. But nevertheless, the embryological development of it has remained stable, if you like, in that it's all occurring within an olfactory embryogenic niche at the start. So it's
interesting.
Yeah, it's very, very weird.
Okay, and then can you describe like the morphology of the GnRH neuron?
So if you can imagine a neuron that has to migrate through a brain. So a brain is a densely packed, organized, highly organized structure with corners and bends and hard bits and soft bits. So the GnRH neurons have taken up this morphology of being an incredibly elongated cell. So it's like a cell body with a big long process at either end. So classically, what we call a bipolar neuron. As most neurons are very much like what would you say, like a star or a spider or something, with branches in many different directions. The GnRH neurons are these very thin, slimline ones. And so you can imagine that, how that structure is important for them to get into the brain, so that they're like a needle, pushing themselves down through into the brain. And once they actually get into the brain, into their place where they're going to stop, they actually maintain that type of morphology. So it's called a bipolar morphology with these very long processes. So
and you discovered there are synapses. So these processes have characteristics of both dendrites and axons. Is that right?
Yeah. So we were always, we've known about that. The community has known about this unusual shape of these cells for a long time. And a few years ago, we were interested to look at the business end of it, i.e. the axon. Trace the axon going down to this place. Well, I said at the base of the brain. It's called the median eminence. The axon's down into the median eminence because that's where they release the GnRH. And long story short, at the end of it, we were shocked to find that actually GnRH neurons don't actually have axons. And actually, it's this long dendrite process that just keeps on going. And so, if you think about it, the cell body is 10 microns, okay? And this process, this dendron or dendritic process, can run for 4,000 to 5,000 microns down before it reaches the median eminence. So it's I know we have some neurons with very, very long axons in our spinal cords, but this is a remarkably long process that runs all the way down. And it's only when the process gets actually into that median eminence area that I was telling you about that it breaks up into these little short axons with nerve terminals where you actually get the release of the GnRH that then goes down into the portal system. So this is a very, very unusual structure. And the key thing is that actually it retains its dendrite-like properties, as you're saying, Mark, that the synapses and the regulation of a neuron actually goes onto the dendrite. And so this dendritic process maintains these synapses. So that's why we call it a dendron, because not only is it dendritic and it's receiving synaptic input, but also it's conducting action potentials just like a regular axon. So we figured it would be easier to call it something, so we called it a dendron. And actually, to this day, as far as I'm aware, something similar has not been found anywhere else in the brain. But again, when I've told you about how bizarre the GnRH neuron is, it's probably not not that unusual that they have lots of strange, unique features. So,
Yeah.
so this morphology. But I have to say that our work is entirely done in rodents, mice, and rats, and we've yet to really. The community as such has yet to prove whether this actually exists in humans or in other species. So it may yet be a feature that's unique to rodents. We don't know that, but in rodents it's certainly there, and it's a very strange thing.
And then, so these GnRH producing neurons, they don't just continuously release the GnRH, or just transiently, there's kind of these pulses, right?
Yeah. So like I said, the hormone, the LH and the FSH released in the blood is, it's a frequency modulated and amplitude modulated system. So the end organs, the ovaries, the gonads, are reading amplitude as well as frequency. So it's released in pulses. So in all female mammals, in the first half stage of the cycle, there's approximately one pulse every hour. For males, we have approximately one pulse every 3 hours for life. Just carries on going. So if you're going to generate pulses of hormone secretion, then you have to tie this up to a neural system, because a neural network's the only way that you're going to generate episodic activity. So we know that the GnRH is released into that portal system, and it's released in an episodic way, and it matches up perfectly with LH and FSH. So I think a key goal for our field and our community as neuroendocrinologists has been trying to decide then how the GnRH neurons actually generate episodes of GnRH secretion, and that's been a really a tough nut to crack. But I think that we've probably pretty successfully done that within the last 5 or 6 years. So yeah.
Can you describe the method? So could you talk about a recent paper that you had in Cell Reports, where you're studying the regulate? So we got the GnRH neurons, and then there are synapses on those from neurons in the hypothalamus that produce kisspeptin.
Yeah.
Can you describe those neurons?
Yeah, maybe I just give a little bit of background to that, if that is okay. So our field, we were all about the GnRH neurons and the neural inputs into those GnRH neurons. And completely out of the blue in 2003, 2 clinical groups, one in France and one in the UK and the USA jointly, published papers which identified rare human individuals that had a mutation in the kisspeptin receptor. So kisspeptin is a neuropeptide transmitter and these individuals failed to go through puberty and were completely infertile, but were normal in every other way. And I have to say that us sitting there, we were completely blown away by this, because no one had ever heard of kisspeptin before. So until these papers came out, kisspeptin was in the realm of cancer biology, particular breast cancer, metastasis and such like. And no one had really ever thought about it being involved in neuroscience or fertility or anything like that. So 2003, we had this really huge step change in knowledge, which interestingly came from the clinic. We were probably never going to get there as fundamental biologists. It's going to take us a long time going through gene by gene. So, this was absolutely fundamental. And it turns out that, quite incredibly, actually, it turns out that the GnRH neuron operates, as I said, it releases GnRH, but it probably doesn't generate patterns of activity itself. And it looks like there are 2 network pattern generators in the hypothalamus. and both of them use kisspeptin, which is quite remarkable. So we have one network that sits in an area called the preoptic area, and it uses kisspeptin, and it's only in females, and it acts on the GnRH neurons to cause the GnRH surge, that thing I was talking about earlier, where the GnRH surge causes the LH surge, which causes ovulation. So these neurons come into play once every cycle. And then we have the other group of kisspeptin neurons, which is in a different place in the arcuate nucleus that I was talking about before. And those kisspeptin neurons, they turn out to be the pulse generator. So these cells are the ones that are active, every hour. They generate a burst of activity, and actually they transmit that activity down actually to the dendron that we were talking about. So these kisspeptin neurons act on the dendron, and then in response to that, the dendron, we get the release of pulsatile GnRH secretion. So coming back to the issue about the pulse generator, I think the field now is pretty comfortable with the idea that you've got a pulse generator network that's sitting, driving the GnRH neurons and telling the GnRH neurons when to secrete their GnRH. So I think that's probably been a reasonable advance for our field. Of course, having made something like that, you then have the next question, and the next question is, well, how do the kisspeptin neurons manage to generate a pattern of activity, once every hour? So what is the mechanism of kisspeptin neuron synchronization? And so this brings us back to this recent paper, Cell Reports paper that you're mentioning, Mark. So, I wouldn't say that we've answered that question to any great extent. What we have done, I think, is identified what are the key players in terms of synchronization of these neurons. So we feel that there are a few places in the brain where you have what are called central pattern generators. So neural networks that actually can interact amongst themselves as their own group to generate episodic patterns of activity. So, these are commonly found in the brainstem for things like breathing and, things like that just unconsciously continue on. So we think that the kisspeptin neurons are a central pattern generator in the hypothalamus that just sit there generating the synchronized episodic pattern of activity. Now, I would just add, it's not to say that other neurons can't influence their activities to slow it down or make it go faster, but left to themselves, they will just generate this pattern of activity.
And how many of those neurons are there?
There's probably about 2,000 in a mouse brain. Yeah. So, I'd add that there's 1,000 GnRH neurons, but probably only about 500 of them are projecting down to be in control of fertility. So we're talking small numbers of cells here, but from a brain perspective. So we've got a situation where these neurons are forming this interconnected ability to actually generate episodic patterns of activity. And another curiosity, I guess, about our field is that these neurons actually express multiple neurotransmitters. So, as neuroscientists, I think we grapple with, one neurotransmitter and how that interacts with another neuron and all the subtleties and ways that you can modulate that. So the kisspeptin neurons have at least 4 neurotransmitters, if not probably more. So we know that they are fundamentally a glutamatergic neuronal phenotype, so they make glutamate. They also make kisspeptin, obviously, because kisspeptin neurons. They make a neurokinin B, which is another neuropeptide, and they also make dynorphin, which is one of the opioid family of peptides. So we have this this quite interesting range of neuropeptides and neurotransmitters in these cells.
And Allan, kind of historically, we think of, the classic neurotransmitters, glutamate, which is the main excitatory transmitter throughout the brain, GABA, the inhibitory transmitter. And then these neuropeptides we think as neuromodulators. So the glutamate, actually its receptors are ion channels, sodium channels or calcium channels. And so when glutamate binds to those receptors, the sodium channel opens, the membrane depolarizes as the sodium rushes in. And then, but the receptors for these neuropeptides are different. They're not ion channels. They signal differently. So you're kind of looking in this cell reports paper, you're kind of looking at the roles of the main neuroexcitatory transmitter in the brain, glutamate, and these other two neuropeptides, right?
Yeah. So I think the end, probably a simple way of explaining it is that we think that the key neurotransmitter that's enabling all of these individual kisspeptin neurons to talk to each other and synchronize is glutamate. So glutamate acting through AMPA receptors, just as you say, classic ionotropic receptors, very potent, very powerful, very brief excitatory inputs. And what we think happens is that even though these cells synchronize, and they synchronize, so we know from recordings in the brain, we know that they synchronize for a period of about 1 minute, and they do it very intensely, and then they stop. So we have a 1 minute period of synchronization, then they stop, we wait an hour, and then we have another intense period of synchronization. But having said that, we think that the kisspeptin neurons are relatively, actually loosely coupled to each other. So if one neuron releases glutamate onto another neuron, it may or may not activate it. It really depends on all of the other inputs that are going on at any one time. So we think that what happens is that there's like this sort of what's called an emergent network. So there's glutamate transmission going on. It's random, a bit chaotic, but one cell might get a bit more activation, and that might activate another cell, and then you get this exponential activation of all these cells, and they're all going together. So we think there's what is termed in the field as emergent glutamatergic transmission that actually brings all these cells together. So then what happens is that then cells are all firing. They're now firing at relatively high frequency, and that's the time when neurokinin B, this neuropeptide that you were talking about, this neuromodulator comes in, and then we think this adds on top of that, increases the synchronization and just really brings it, completely to the fruition where we get this really massive, sudden, burst of activity. So we think that happens. We don't know actually why the cells stop firing, but we think that it's primarily due to what are called intrinsic mechanisms. So neurons actually don't like to fire very fast for a long period of time because they fill up with calcium, and that's not good for them. So they have a lot of mechanisms that actually limit the self-limiting mechanisms that limit their own firing. So we think that these intrinsic mechanisms are the key thing that actually then stops all of their activity, and they just calm down again. So we have neurokinin B as you said, Mark, as a classic modulator of glutamatergic transmission. I mean, that's sort of pretty standard. I think what's probably more interesting, and to be honest, the one we know a lot less about, is dynorphin. So it looks like dynorphin is really setting perhaps the scene for when that glutamatergic immersion activity can occur. So dynorphin is an inhibitory opioid transmitter, and it looks like it's involved in actually setting how easy it is for the glutamate transmission to break through and become an emergent network. So if you have high levels of dynorphin tone, then it's rather difficult. So you don't get synchronizations very frequently. But if the dynorphin tone is low, then the glutamate's free to just generate these emergent activities all the time. So I mean, that's our understanding on the moment. No doubt that will change and be refined, but it's a model that we've been able to put forward. So that's essentially what that recent paper is about.
And then so estrogen can affect this process?
Yes. So we were, to go right back to the start, we were talking about how the ovary is producing estrogen and progesterone, and how it's very important that that feedback occurs, back. So we have the homeostatic, feedback cycle. So it's very important that the pulse generator knows what the physiological state of the ovary is. So the estrogen and progesterone, they feed back onto the pulse generator and basically slow its activity. And just to put things into perspective, I said before that, that there are other inputs that will come into the pulse generator that can slow its activity down a bit or increase its activity, depending on the environment and the status of the animal. Without any question, the most important modulator of the pulse generator activity is estrogen and progesterone. So they have a massive inhibitory influence, as you'd expect, and it's a homeostatic circuit. They have a massive inhibitory effect on these cells. So I said that the pulse generator operates, once every hour. If you take away the ovaries, you take estrogen and progesterone away, then the pulse generator essentially just goes a bit crazy. So it'll start going every 15 minutes. And instead of that period of activity, I said it goes for 1 minute. It'll go every 15 minutes, and actually its period of activity can last for 10 minutes. So it goes without these gonadal steroid feedback. It's in an extremely unstable, I think that's the best way of putting it, unstable pattern of activity. So it's firing off all the time. It's difficult for all that synchronization activity to stop. And then when it stops, it starts again very quickly. So gonadal steroids are really, really very important. So
and testosterone and testosterone will also have similar effects.
It's exactly the same. Yeah. So
and are those effects of testosterone mediated by testosterone itself, or after it's converted to estrogen?
Yeah, a good question. And I don't think we've got a definitive decision for that. We would mostly think it's after what you're talking about is the aromatization. It's after aromatization to estradiol. But studies have knocked out estrogen receptors and androgen receptors and kisspeptin neurons. And well, the problem with those studies is you get a lot of compensation. When you take one thing away, then another thing can operate. So certainly estrogen and androgen can both work in males, but the relative balance, again, I don't think we know the answer as to that.
And how early during development? So is all this going on before puberty? And in the the GnRH neurons and kisspeptin and all that?
Yeah. So in terms of the pulses and the pulse generator, that is probably active in utero. So that whole network is up and going in utero. What happens is that it then, quite soon after birth, it just completely shuts down. And so it shuts down, and then we have this sort of, the sophisticated scientific term magic, which by some magical process, it all restarts up again for puberty. So the pulses then start up. The pulses, you can imagine, go down, and they get the ovary going again. So the ovary starts up again. The ovary starts making estrogen and progesterone. That feeds back on the brain. And then in females, the surge network gets going. So the surge network comes after the pulses. So, the pulse system is way there early on.
And there's no, you mentioned the mutations in kisspeptin, and the effect on the development of the GnRH neurons. Has people done manipulations of neurokinin, dynorphin, in this pre-puberty I guess the question is there a way you can turn this on earlier? Reinitiate. Yeah.
Yeah. So if we look in the clinic again, there are rare human beings who have mutations in the kisspeptin receptor, which means that the kisspeptin receptor is present out on the membrane of the cells more than it's just very stable, so it doesn't get degraded very well. And in these individuals, they have precocious puberty. So these people having puberty are starting at 5 or 6 years of age. There's also an interesting study by the Sukumura group in Japan, and they had a rat model, and essentially they rescued kisspeptin expression. So they had animals that had no kisspeptin, then they gave kisspeptin back. And when they gave kisspeptin back to these animals, within 4 or 5 days, I think it was, the whole puberty process started up again.
Yeah.
So I think, and obviously in individuals that have no kisspeptin or no kisspeptin receptor, they never go through puberty. So it's pretty clear that the pulse generator that I was talking to you about is a kisspeptin. It's the same thing. It's a kisspeptin dependent process. The roles of dynorphin in puberty are almost unknown. Some work has looked at the other transmitter, NKB, and I think it would be fair to say that NKB looks like it's important actually in that whole pubertal process. But then, well, that's exactly as we would expect, because NKB is important in getting the final kick to those synchronizations to get things up and going. And remarkably, and perhaps because we haven't been thinking about glutamate as much as we should have, there's probably very little actual real hard data on role of glutamate at that time in the puberty process.
The onset of puberty can kind of, in a crude way, but maybe evolutionarily, not maybe, almost surely evolutionarily important, is energy intake, right? If you severely restrict energy intake, it actually can, delay the onset of puberty. If you overnourish animals, they have an earlier onset of puberty. And I think in humans, there's evidence too, right? I think there's even some evidence that there's a role for childhood overweight or overnourishment in an earlier age of onset of puberty, at least in females.
Yeah.
And is it known how, and now we're getting in a whole new area, but is there any insight into how this energy intake affects these systems that you've been studying?
I think a good framework to think about this is that, and perhaps not in humans, but in animals, from an evolutionary perspective, and from a female perspective in particular, you really don't want to be reproductively competent unless you're in the right internal and external environment. I mean, you need to be healthy within yourself. You need to be in a non-stressful environment, all of that. I mean, that's a principle that even works for later on, actually post-puberty, when you have situations where ovulation doesn't occur and such like. So there's a lot of good reasons for having, if you think about it, multiple little, checkpoints along the way for these neural processes to occur. So, am I under little stress? Yes. Do I have no infection going on? Yes. Do I have enough, body reserves? Yes. So I think that the concept, at least my concept, is that the development across puberty, of course, in humans, this is, takes 4 or 5 years from start to end to finish this process. Of course, it's quicker in other animals. But it's a gradual process in which you get the maturation of this pulse generator. So, the key event you want, let's say, is to get your pulse generator going once per hour. Once you've got to that point, then you're going to generate the ovarian function and the steroids that, will get you there. But at the start, it's, as I say, early on in puberty, it's probably not operational at all. So you've got a slow process by which the pulse generator needs to receive the information telling it, yes, this is okay, yes, this is okay, yes, this is okay. We're now at the point where we can operate. And possibly one of the better ones that we would understand is the body weight one, in the sense that it looks like leptin, fat, body fat makes a hormone called leptin. So leptin circulates in the body. So the more body fat you have, the more leptin you have. And so I think, again, a pretty convincing case has been made by the community that leptin levels need to reach a certain threshold for the whole puberty process to start. So it's not actually the point that if you get to this point, puberty will start. It's if it gets to this point, then in a sense you've checked off, yes, I have the body resources, to go on and be pregnant. I have that. So in that way, it's very often leptin is talked about as a permissive factor. So we have leptin operating on this network. I mean, one of the key questions, which I think is still unresolved, and there's perhaps some argument about whether the leptin acts directly on the kisspeptin neurons or whether leptin acts on other neurons, that are associated with kisspeptin neurons to generate that. So that would, I think, be my take on how we can think of body weight being like that. So, an individual who's not got the body weight is not getting leptin up into the correct level area. And so the whole puberty process is just struggling. It's saying, where's the signal? Where's the signal? And in fact, even without the signal, the other ones cumulatively can push it over the bar into puberty, but just not going to occur at a normal time. So I mean that's puberty, but then there are other directly energy sensing molecules that are in kisspeptin neurons themselves that are probably important for regulating kisspeptin gene expression and things like that. So I don't think the nutritional thing, what you say is absolutely correct. Nutritional imbalance either way has an impact on when puberty occurs, but I don't think it's going to turn out to be one simple mechanism, and it's probably multiple sensors, maybe even looking at multiple different nutrient composition nutrients. So
so we're getting towards the end here, I think, Allan. I have two final questions. What do you consider your most important discovery? You have to pick one.
If I have to pick one, probably it was I think it was providing the definitive evidence that the kisspeptin neurons in the arcuate nucleus were the pulse generator. I mean, that was cracking a pretty fundamental thing. Where is the pulse? What generates pulses? I think, if we have to go for one, there you go.
Okay.
I've never been asked that before.
Okay, well, we'll see if you've been asked this one. What's the most pressing unanswered question you're trying to answer now?
Actually, we've become very interested in puberty. I mean, puberty, the mechanism, the fundamental mechanism, brain mechanism that which puberty is generated has remained a real mystery for a long, I remember, I think it was in 2005, Science came out for its 125th issue or anniversary. It came out with its 125 great questions of, unanswered questions of science. And it was, how was puberty initiated? And, and we just still don't know that. So I think that now that we have some mechanistic insight into the pulse generator how it's working, how these cells are synchronizing, I think we might be able to make some traction. So I think that's something that we're really very interested in at the moment.
So you need to do some, you do obviously a lot of studies on pre-pubertal, and before their synchronized activity occurs. I have two questions. They're just kind of as scientist to scientist. So, you've developed this it's called acute brain slice preparation, where you find you essentially have a slice of this hypothalamus with both the kisspeptin neurons and GnRH neurons in it. And then you use this elegant method of, it's a little complicated, but it's a really beautiful method now of, you can actually look at them under fluorescent microscope and get an indication of the changes in calcium level. We did work with what are called organotypic slice cultures, where you can maintain them for, we could maintain them for a month or more. Now this is hippocampus, and we'd still get kind of the synaptic correlates of learning and memory and all this. So if I had a postdoc in the lab, I'd say, well, try to maintain these hypothalamic slices for a month, and then look at the calcium activity during that month. But the other way is in vivo. Of course, in vivo, it's better because it's the real deal, right? But sometimes it's a little harder to do the experiment. So I guess, kind of summary, how are you approaching this technically to look at these neural signaling systems, before puberty?
The news you don't want to know is that Kunjum Kim in Korea has already made primary cultures of the arcuate nucleus and looked at the kisspeptin neurons and looked at their synchronization and their synchronization behavior and how they may interact and such like. And the thing that you probably won't want to hear is that the activity patterns, the behavior, the neurotransmitters that are critical are not the ones, are completely different to what you find in vivo. So hence, we come down to that problem with any culture type arrangement, that it can just reorganize itself and generate lovely patterns of activity that are
but once it's established, if you made hypothalamic slices from whatever 2-week-old animals, which is a couple of weeks before the onset of puberty, I think. I don't know. I would predict that circuitry would be established and might be maintained.
So I think, so I just want to raise that in terms of long-term culturing of cells. But our approach is going to be using the acute brain slice procedure because
so just do it at different times.
Acute brain slices, we can look at it over developmental ages. The challenge is that in vivo, we've been lucky to be able to harvest these incredible techniques off the neuroscience community that allow us to look at activity of neurons in vivo with animals walking around their cages. But this does require a certain degree of surgery, and these animals have a certain amount of headgear on top of them. We're trying to push back the time at which we're able to start making these recordings in mice. And we're in the preliminary stages of doing that, but we are effectively actually measuring the pulse generator activity before puberty onset now. So
good.
We can't go way back, but we can go back probably far enough at the moment. So
Okay, good. Well, I hope you have a good 2023.
Thank you very much. Yeah.
And, you know, I'm actually, my background is in neuroendocrinology. I did a master's degree where I was studying cultured adrenal cortical cells that they don't produce cortisol as their end product, but they produce a different steroid. And I was looking at how cortisol itself affected the cells that produce cortisol.
Right. Yeah.
And I found it did. And then my PhD work was on a neuroendocrine system that controls molting in crabs, which there's a neuropeptide hormone that's released into the blood, and it affects the gland that produces the molting hormone. So, you know, I'm kind of returning with this podcast to some of my previous, you know, because it's been a long time since I thought about, you know, neuroendocrine systems much.
Yeah.
Okay, Allan. Well,
thanks for hosting me on this, Mark. Thanks very much. Yeah.
Okay.
Okay.
Take
All
care.
right. Goodbye from
Bye.
the UK. Yeah. Bye-bye.
Bye.